Good morning. Good morning, everyone. We're going to go ahead and get started. Today is a jam-packed day, so we want to start as close to 8 o'clock as possible. Before we get started today, I'd like to do a big thank you to our NUCDF board members, which none of them are in the room. They are all working. I don't think there's one of them in here. Eva Oslin, Lori Shockey, Tony Martinez, there she is, Tony. Amanda Sonnentag, she's running, and Mary Alice O'Malley. Our board is the longest serving board in rare disease. Most of our board members have served more than 20 years. So help me thank them for their commitment. I'd also like to give a big thank you to our medical advisory board members. If they could all stand, I'm sorry, they're all eating. If you could stand, our medical advisory board members, thank you so much. Your commitment to the UCD community is why we are able to stand here today and to provide this resource. We have many of our clinical researchers from the UCDC in the room as well. If all of the, anyone associated with the Urea Cycle Disorders Consortium, if you could also stand. They are the reason that we have the treatment options, the research outcomes. It is because of them, and we honor their commitment. I'd also like to give a big thank you to our sponsors. You'll see outside we have our gold sponsor, which is Ultragenyx, Zevra, and Amgen. Our silver sponsors, which is Mendic, Eton, and Arcturus. And our bronze sponsor, which is Nutricia. They are out and in the hall. Please visit their booths and let them explain their options to you. And we're grateful for their commitment. I know that in the room we have many old families and new families. Those of you that are old families, you are our legacy. You are why we have what we have today. All of the new families, you are the legacy. We want you in 20 years to be the reason why we're still here. So thank you for everyone. I'd like to tell you just a little bit about my family. Our legacy started when our daughter, Lexi, which is here, was born in 1997, and she was diagnosed with OTC at 14 months. We were desperate for answers. We called Cindy Lamons with NUCDF. She took us under her wing and said, come to a conference. We came to a conference. We found our community. We found our family. We found the best doctors in the world. And that's what kept her alive. And it's because of that that she is a vibrant 26-year-old living her best life. So she is our legacy of hope. I've been to every conference for the past 25 years. I came and got hooked. And I know that there are families in the room that have been coming for at least a decade. Could those families stand for me? Thank you. And I know that there are brand new families in the room. If this is your first conference, if you could stand, please. First conference. Okay, our veteran families, that's your homework. You saw them. Take them under your wing, bring them into the fold, bring them into the community, help them to make the best options for their family. Before we get started, I'd like to acknowledge two of our staff members. One of the things that we have done as a board, as we have done strategic planning, we are laser focused on our mission, and we knew that building the infrastructure of NUCDF was going to be vital. NUCDF was Cindy, and we had Cindy and our board. And we knew that it takes about 4 people, maybe 5, to be Cindy. So we knew that building the infrastructure was going to be very important. If you have seen our social media or our website, you see that we have hired someone that's an expert. Her name is Amanda Swank. She came on as an administrative assistant, has turned into a social media expert, and has just been completely invaluable to us. So we're really happy that Amanda is with us. And then we have our Deputy Director, Jill Williams. Jill came to us in August, and it's been the best thing that we've done. Jill has a family member that's affected with urea cycle disorders, so she has a depth of knowledge about urea cycle disorders, but also about research and also about communications. Jill has been wonderful, and I'm going to turn the day over to her. So a big thank you to Jill, and we'll get started.
Thank you, Teresa. I want to extend a huge thank you to Teresa. She stepped in and picked this organization up and has kept it moving forward. And without her, this conference would not be happening. So let's give a thank you to Teresa. So I first heard the words urea cycle disorder 29 years ago when my nephew was diagnosed with citrullinemia. We had no idea what that would mean at the time. And I have watched him and his family for 30 years figure out how to navigate this world. I am grateful for the support that they received from NUCDF, from other families, and from the UCDC experts. So last August, I was thrilled when Teresa asked me to join NUCDF staff, and I hope to contribute to the next chapter in NUCDF's history. I'm going to be sort of keeping the trains running on time today and announcing speakers as they come up, but we're going to start off with a few announcements. So first, and maybe most importantly, lunch is at noon. It's going to be in the Ceriso restaurant upstairs. You should go straight up the escalators, turn left, you'll find that. If you have children in the child care room, please plan to pick them up about 10 minutes early at 11:50 so that the child care folks can also have lunch. For the bathrooms, go down this hall, turn right, they're at the end of the hallway on the right. The sessions today are being recorded, and they'll be made available to families after the events, so don't feel like you have to write down every word that people are saying. We're going to do our best to keep the schedule running on time. We're going to save time for questions at the end of every talk. So if you want to ask questions, we are asked that you're going to come to the middle of the aisle. We'll have microphones. We want to be able to record your questions. If we run out of time for questions and you don't have the chance to ask yours, there are going to be panel presentations, and that's going to be 30-minute opportunities to ask more questions. So just jot them down and save them for the panel. If you have questions, please keep them general. We want everybody to benefit from the answers, and the doctors are not able to provide specific medical advice. We want to keep your private information private. So we're going to open today with a keynote address called Legacy of Hope. Then you're going to hear a series of talks from several experts featuring the latest insights on UCD care drawn from their research findings. And we'll have 2 panel discussions this morning. There we go. Now I'm pleased to introduce Dr. Sandesh Nagamani as our keynote speaker. He's an associate professor at Baylor College of Medicine and director of their clinical research division in molecular and human genetics. He's a key member of the NUCDF's Medical Advisory Board and one of the first people we call when we have questions. He received the NUCDF Fellowship Award in 2011, which helped fund his research on nitric oxide and ASA deficiency. And he shared yesterday that without that fellowship, he may not have continued working in the UCD space, or in the urea cycle disorder space. So we're grateful that Cindy's fellowship legacy is going to continue bringing new doctors into the fold. Dr. Nagamani also serves as co-principal investigator of the Urea Cycle Disorders Consortium. Dr. Nagamani, thank you for joining us this morning.
Good morning, everybody. So I'm extremely grateful because NUCDF not only gave me a fellowship at the critical time of my life, but also I see that they've given me a PhD, which I did not earn. So I am running with it. Y'all all heard it here, and I'm MD PhD from now. And Lindsay Birch cannot make fun of me that I don't have a PhD again. So thank you. So I think my job is easier today because you're going to hear from a range of specialists who are going to talk to you about specific topics. I just have to give you a 20,000-foot overview of what the NUCDF does, what patients do to a field, and what patient advocacy does. But before that, welcome to Houston. This is our home, and though we're 35 miles away from Houston, Woodlands is not Houston, but we'll not hold it against them. If you guys are staying the weekend, there are multiple things to do in Houston. There are beautiful parks. The weather is going to be nice. There are tons of museums in the museum district, and for the children who are interested in dinosaurs, the museum in Houston has one of the only 3 Triceratops skeleton intact across the world that's ever on display, so it's cool to go and watch that. For space enthusiasts, we have NASA. If you have not gone, it's quite a drive from here, but that should be on your bucket list of things to do. And as a city, we're very proud of the restaurants and food we have, so please enjoy the restaurants in Houston as well. These are my disclosures of interest. I do collaborate with pharmaceutical industry partners in some research, but nothing I'm going to talk today is related to my work with partners outside of academia. And also, if Cindy were here today, she would not have let me put this slide up. And for those of you thinking, how did he get a single photo of Cindy? I did not. I had to crop out Teresa Warner and Amanda from this. And you see Amanda's hair still there. So today is Cindy's birthday. And so I think it is most appropriate that the conference is called Legacy of Hope, because a lot of what has been achieved within the field we do owe to Cindy. So how I thought I'm going to fashion my talk is just to provide a very brief historical perspective of the discovery of urea cycle and the urea cycle disorders, tell you where we were as a field with urea cycle disorders decades ago, what are some of the challenges that we, both in the medical profession as well as the patient community we have, in improving outcomes for UCDs, and I'm going to specifically talk about the role of patients and patient advocacy groups. And I'm going to talk about collaborative research networks that move the field forward to improve the outcomes. Finally, I'd want to end by showing you how the outcomes have improved. I'm not going to go over the data, but give you a flavor for where we are and where we have to go from here, fitting into the theme of Legacy of Hope and how all the rest of the talks fit into that. So about the urea cycle, all living things need nitrogen. Nitrogen is an important ingredient for building blocks called amino acids that are present in all of our proteins. It's an important ingredient for nucleic acids that form the genetic information. So all living things need nitrogen. There's nitrogen everywhere in the atmosphere. Nearly 80% of the atmosphere is composed of nitrogen, but there's not one atom of nitrogen that we breathe in that can get incorporated into our body. So none of what we breathe gets into our tissues. All the nitrogen that we get comes from our food sources, either plants or animals. They're broken down, and then they make the proteins, nucleic acid, and so on. The thing with nitrogen is that after nitrogen is used to build the important building blocks, there's not a good storage form for us to store it. Like we can store fats and we can store carbohydrates, which are converted to fats within our bodies. So the extra nitrogen, after we've used it for proteins and other functions, has to be excreted out. And in the matter when it has to be excreted, it forms a compound called ammonia, which I don't need to tell any of you is a toxic compound. So how do we get rid of ammonia? It depends as to who you are. If you're an aquatic animal living in water, ammonia can just diffuse out, and you don't need a specialized way to sort of discard this ammonia. Now, ammonia is not very soluble in water, so it's not a problem for fish. What happens when animals evolve to live on land? We don't swim all the way around, so we can't end up disposing ammonia off. So evolution made a different way of disposing this ammonia. So if you're a bird or a lizard, and my kid has called me that once, you produce uric acid, a compound that has nitrogen, and you excrete it out. If you're a mammal, and i.e. human, the only way you can excrete out excess nitrogen is by forming this compound called urea. Now, in amphibians, like frogs, when you're a tadpole and you're swimming all the way in the water, you don't have a urea cycle. Your ammonia goes out. But once you become an adult frog, you actually need the urea cycle. So how is it that urea is produced? How do we convert the nitrogen from ammonia to urea? And this was a key biochemical discovery in the 1930s. Shown here is Dr. Hans Krebs, a biochemist he was German and he was British. He was using liver slices to study metabolism. And in the early 1900s, it was known that if you take this amino acid arginine, and I know many of you might be on arginine, and you put in a bacterial enzyme that can cut arginine into two compounds, you have ornithine and urea. So it was known. So in the liver slices, surprisingly, he found that when you add ornithine, the urea went up. It didn't make sense. He could make out that if arginine you put, you have more urea because that's how it is split. If I'm adding ornithine, why is urea increasing? What he also figured out is that on the liver slices, if you sort of put ammonia or you put carbon dioxide, even then the urea increased. And being the biochemist he looked, no matter whether you put ornithine or the ammonia, only urea increased, but the ornithine never increased. So he thought, you know what? Maybe this is a cycle. The ornithine never gets out and gets stuck inside the cycle. So he figured out that the next step would be citrulline, and he thought that another ammonia gets added to form arginine, and so this is how all mammals synthesize urea, and this was called the urea cycle. Now, there are other cycles that we have organisms have, but this was the first cyclical or cyclical metabolic cycle ever discovered in organisms. I know Dr. Bird spoke to you about the urea cycle yesterday, but I'm just going to go over it briefly. So the purpose of the urea cycle is to take nitrogen from the toxic compound ammonia, shown up there in the red, and pass it on through a series of sort of compounds, which are all in black, to trap the nitrogen and finally convert it into urea, which is in green, so that it can be excreted out. Think of this as a Lego block. Nitrogen is one Lego block, but you have to join it with other Lego blocks, and then you take out the Lego blocks, and finally you have the right sort of Lego block, which is urea, which you can excrete out. So you need a lot of enzymes and transporters to do this within the liver. Whenever there's a block in this traffic, you cannot dispose of this waste ammonia, and ammonia goes up. That's what gives rise to urea cycle disorders. Now, depending on where the block is, you have different urea cycle disorders. If you have a block in the enzyme called OTC, you have OTC deficiency. If you have a block where the enzyme ASS has to join two Lego blocks that are citrulline and aspartate together, you have what is called ASS1 deficiency, but again, because the citrulline can't go through the cycle, it accumulates, and there's a lot of citrulline in the blood, so that's why it's also called citrullinemia. Similarly, if the block is just one step down, you have the compound ASA, argininosuccinic acid, accumulate, That's why that disorder is called argininosuccinic aciduria. And if you have a block that splits arginine finally to urea, then you have a high amount of arginine, and that is called argininemia. Now, though the urea cycle was discovered in the 1930s, it took another 20 plus years, 25 to be precise, for a first human with a urea cycle disorder to be described, and that was argininosuccinic aciduria in a child who had intellectual disability. The physician saw that she was excreting a lot of an amino acid compound, which was later found to be ASA, and this was the first urea cycle to be discovered. Within the next 4 years, the second UCD, which is OTC deficiency, was discovered in someone with hyperammonemia. And over the next ensuing years, citrullinemia or ASS1 deficiency, argininemia or ARG1 deficiency was discovered. So and CPS1 deficiency were all discovered. So within the first decade of the first enzyme deficiency, the other 5 were discovered, and the final enzyme deficiency, NAGS, which is extremely rare, was discovered in another 10 years. Now, these are all the urea cycle disorders. The primary urea cycle disorders can be the enzyme deficiency, which are the enzymes listed there. They can be transporter deficiency, which have to move the Lego blocks through the traffic. The 2 of them are described there. And you also have other, as Dr. Burge would have mentioned, traffic that has to enter into and out of the urea cycle. So if you have disorders that affect that, you have secondary urea cycle disorders. So the term urea cycle disorders encompasses different types of these disorders. Even though the first urea cycle disorders were discovered only in the 1950s, within the first 20 years, physicians and scientists had come up with ways to decrease the flow through the urea cycle. Diet, amino acids were early treatment modalities, but by work from Saul Brusillo and Mark Batshaw, who was one of the founders of the Urea Cycle Disorders Foundation, also introduced this alternate pathway therapy. So the idea was, if your main loop, the 610 loop for Houston, is blocked and you have to get from point A to point B, can you take one of the alternative paths? Can you take a local road that bypasses this? Thus, the traffic block within 610 is decreased. So the alternate pathway with either benzoate, phenylbutyrate, and glycerol phenylbutyrate, all of those come from that idea. And by the late 70s, the first alternate pathway therapies were just becoming available, though not widely. Before such therapy became available, these were really devastating disorders. And in a paper by Dr. Shi in 1976, when they reported on 28 children with urea cycle disorders, the 1-year survival was only 14%, and 75% of all survivors had intellectual disability. Even within a short span of time, at the Johns Hopkins Institute, where the alternate pathway was being used, when they looked at children who were being treated, the survival was significantly better, but majority of the survivors still had intellectual disability, and the average IQ was still in the 40s. So one of the challenges in advancing care and outcomes in urea cycle disorders are that the natural history of the disease is not known. We don't know what organ systems they affect. We don't know what treatments work, what treatments do not work. Rarity also means that there are very few specialists who are trained to take care of these disorders. There are few expert centers for research and treatment, and conducting clinical studies usually becomes difficult. So many a times for many genetic disorders and many other rare disorders, we often do not have high-quality evidence to guide therapy, and we do things that we think are physiologically reasonable. So if we ask ourselves, what are some ways in which we can improve the outcomes for rare disorders like urea cycle disorders? There are many ways. I've just broken it down into 4 sectors of a circle. In the first sector, what drives any advancement is research. It will be laboratory research that identifies new targets for therapy, clinical research that implements such targets for therapy, and collaboration with industry partners who make such therapy feasible from an implementation standpoint. It will be clinical care, improving the diagnosis, such as by newborn screens, instituting appropriate therapy, and getting appropriate services and referrals, referring to liver transplantation when required, and improving access of care will improve outcomes. Public policies are a cornerstone of this. Newborn screening programs that allows for early detection, the Orphan Drug Act that encourages the industry partners to develop therapies, and from a practical standpoint, when such therapies become available, coverage of the agents via insurance so that it's widely available to all individuals at an affordable price will improve outcomes. And more importantly, families, health caregivers have to be educated because these are rare disorders, and we have to train the next generation of physician scientists who will continue to care for individuals. We're all so great at doing things in our own silos that we do not know what the others are doing, and many a time this sort of siloed work impedes progress because the right hand doesn't know what the left hand is doing. And this is where, to tie all of them together, we need all of you, the patients, to tell us what is important, and we need patient advocacy groups like the NUCDF to keep us all in line and say, what do we need to do and where do we need to go? So what are your roles and the patient advocacy roles to improve outcomes? It is to inform researchers about what is important, a patient-centric research. It is to participate in clinical trials and inform other people of what the pluses and minuses of any clinical trial are, and to advocate for research to make sure that it is important and it moves forward. On the clinical realm, again, a lot of the physicians, what we learn is from the advocacy groups and the patients. It is important for you all to educate your healthcare providers and be advocates for yourself. You're the champions that make public policies happen. Yesterday, when Teresa was trying to tell us how she had to fight with the Woodlands city to let the lanterns flow, that approach has been taken to make sure that the governmental agencies and the insurance also don't place roadblocks and make all of the treatments and diagnosis available. So that's an important part of patient advocacy. And finally, you all have been doing this for a long time, educating providers and training the next generation of scientists also is an important thing from the patients. There are many patient advocacy groups that are listed here. Many of them are nonprofit and do good work. The one that I'm closely associated with and the premier advocacy group for the UCDs is the National Urea Cycle Disorders Foundation, and I will talk about a couple of things that they have done as to how it has advanced the field. So the NUCDF was set up in the late 1980s, 88 to be exact, and within the first couple of years, families were already invited to participate in this foundation. In the early 90s, when sodium phenylbutyrate, or buphenol, was not widely available, and it was only being produced and compounded in the Johns Hopkins pharmacy, and clinical trials had to be done, there was not enough funding. NUCDF championed funding that allowed for the clinical trials to move forward, that allowed for the first drug to be approved for the disorder, buprenorphine, in 1996. Without the advocacy of the NUCDF, that would not have happened. In the early 2000s, the NUCDF also took a leadership role in advocating for newborn screen. They partnered with other broader advocacy groups to perform rare diseases advocacy. You heard yesterday about NUCDF having the first treatment guidelines, in the early 2000s that came out. And because they had a legacy of doing this for almost 2 decades, when there was an opportunity to form a network for urea cycle disorders, and the NIH was interested, the National Institutes of Health was interested in funding it, NUCDF became a primary partner that established the Urea Cycle Disorders Consortium with many of the educational institutions, and some of the leaders that did start this are sort of sitting in the audience today. The NUCDF was also pivotal in getting philanthropic funding with the O'Malley Foundation that allowed for expansion of this network, that allowed for collaboration and providing advice to industry partners on how to do the drug development. The approval of Ravicti or glycerol phenylbutyrate would not have happened without the advocacy from this organization. And the NUCDF continued their lead roles in educating people, in hosting international consortiums, educating people about ammonia checking, to do a comparative study of liver transplant versus medical management, and the principal investigator for that, Nick Amu, is sitting in the audience today, and so many more educational activities and advocacy activities. So the NUCDF has been involved all the way from starting from drug approvals to initiating an expansion of networks to educating the community about the disorders. And the consortium that is dear to my heart, because I did grow up in this consortium, is the Urea Cycle Disorders Consortium. And the members of the UCDC who were recognized a few minutes ago are located at multiple sites. And what this consortium does, and we do have a table outside, Jennifer Seminara is sitting in. She is looking at what you would want for the, Jennifer, do you want to just stand up so that people know? So Jennifer has a table. She's got a QR code which will say, what do you as families want to hear from the newsletters? So please go and reach out to her. What the UCDC does is to do clinical studies and research, and listed here are 18 studies of the UCDC, all the way from the natural history study to clinical trials that have looked at the treatment modalities. Many of you and your families participate in these studies, and thank you for that, and we would not have been able to do any of these without your participation. Now, while I know of only I've worked closely with the Urea Cycle Disorders Consortium, I also want to acknowledge that there are many other collaborative groups which are listed here. The European group, EIMD, the Citron Foundation group, which runs out of Singapore and Japan, as well as Dr. George Mazuragos will talk to you about the Society for Pediatric Liver Transplantation and UNOS, and also multi-center collaborative efforts without a formal structure. All of these have pulled us out of our silos to be able to work together to answer important questions. So with such collaborative work in the US and elsewhere, there has been a significant increase in the number of medical articles that are being published on UCDs. If you were to look at this, in the 1970s to 80s, we had only hundreds of publications worldwide. Now we touch close to 1,000 per every decade. So this is not only about publications of the articles. With all of these collaborative networks and the understanding of the disorders, there's a lot of interest in the pharmaceutical industry as well to develop new therapies. Now, for those of you who want to know what studies are available, there is a site called clinicaltrials.gov, which is a governmental site because of the .gov, and it lists all the clinical studies where you can search by condition. You can search, you can refine your search by location, by whether they're enrolling children versus adults, who the sponsor is, and what it entails. So this is a good site for you to go and look into it. The reason why I brought about clinicaltrials.gov is, along with the medical articles that have seen a significant increase in UCDs, there has been an increase in the number of studies as well. In clinicaltrials.gov, if you look, there are a total of 77 studies, but using the term urea cycle disorders, out of which nearly 51, over 50 are treatment-related disorders. So this is something you have to go back to your physician, your trusted care providers, and ask whether these sort of studies would be relevant to you, and they would be safe for you to do. And I think you're going to hear from a few of the other individuals about this. So with all of what has happened, where are we now as compared to the 1970s, where the outcomes were not good? This is a paper done recently from the EIMD and UCDC, and if you were to look at the cognitive outcome, on the vertical axis is an outcome measure, and you see small numbers, 2, 0, -2, and -4. And if you're in the green shaded region, that means that the cognitive outcome is what one would think is within the realm and of being normal. And you look that, as compared to the 1970s, a significantly higher proportion of individuals are doing better currently, whether it is the ASS1D, ASLD, or the OTCD. This does not mean that we don't have challenges or improvements to make. So how do we further improve the outcomes for UCDs? From a diagnosis and monitoring standpoint, we need better screening techniques, newborn screening, genetic screening, better genetic testing for earlier diagnosis. And the more genetic testing we are doing, it is becoming harder for us to interpret some of the variants. How do we know which is disease-causing versus not? Dr. Amy Dudley is going to talk about her yeast genetic studies about that. We need better monitoring to look at the neurological outcome. Andrea Grobman is going to talk about the neuromonitoring and MRI imaging. How do we monitor for long-term complications and liver disease? And Lindsay Burridge is going to talk about that in today's session as well. We need better dietary therapy and a regular dietary therapy. Erin McLeod, who's a dietitian, is going to talk about that. We need to make better informed decisions when to continue medical therapy and when liver transplant is appropriate. Nick Amu will be talking about that. And practical aspects and the data on liver transplantation in UCDs, Dr. George Mazarogos will be talking about that. We need newer therapies, which are novel. We are going to hear from our industry partners for the meeting about some of the newer therapies they are involved with. Dr. McCandless is going to talk about genomic therapies in UCDs, and Dr. Berry is going to talk about what it means for you as patients to participate in these sort of studies. Finally, we'll need to continue educating the providers by the patients, and education of patients and families by the providers. That's what the discussions and the sessions, the rest of them for today are. I wanted to thank the entire team at Baylor College of Medicine, our metabolic geneticists who refer patients for the Urea Cycle Disorders Consortium, the UCDC team, starting with Dr. Brennan Lee, who spoke yesterday, Dr. Lindsay Burridge, who's going to talk today, and Saima Ali, who basically is the engine that makes us all look good. If you take Saima away, I would not know whether my right from the left when it comes to UCDC. We're also lucky to work with a lot of neuropsychologists, hepatology and radiology colleagues, as well as past members of BCM who've all been participants of the UCDC. And I want to thank and acknowledge the UCDC team. This is what a collaborative consortium looks like. It has numerous members who do different things, but all equal partners. And at the top of that, I would want to put NUCDF and the patients, because without y'all, the UCDC would not have been able to do what it has to do. And I want to thank our sources of funding that allow us to do such collaborative network, the O'Malley Foundation, Kering Foundation, and other philanthropic organizations that make it possible for us to do the work. I know I'm 2 minutes over the time limit, but please allow me to thank two people. I wanted to thank Cindy Lamons, who, as you all have heard, has been a tour de force to carry all of this forward. Cindy took a chance on me when during my earlier days as a researcher, and that made a significant impact, and I learned a lot from Cindy of what it means to be in a rare diseases field. Teresa Warner would not want me to put this picture up, but I did not acquiesce to that request. She takes UCDC meetings from her school building. Sometimes we hear the kids in the background. In spite of that, she's laser focused to do stuff. She takes calls past late hours. Any of us have to be awake, and yet she's at school every single day. So thank you, Teresa, for doing what you do. And to be honest, I just did not want to put Teresa's picture there, but I put it because of Lexi Warner. I wanted to thank 4 individuals here who I have known since they were yay tall, but now they're taller than me. Lexi spoke to me for 15 minutes. Thank you. John Oliphant was too cool to talk to me. I did not take offense, but still thank you. Andres K. Bosi came and gave me a hug yesterday. Thanks for that. And I don't see Sophia here. And Sophia, thank you for correcting my spelling. Thank you very much. And I'm a couple of minutes over, so I think maybe it would be best if we hold the questions for the after the talks. Thank you.